What's up, TUMNAD lectures? In this video, we are looking at what we call chemical digestion and absorption of what we call carbohydrates. You find it helpful at the end of it, or please make sure to like the video, comment down in the comment section, and then subscribe. Let's go ahead and get started. In this video, we are going to be mainly focusing on the basics of what we call chemical digestion and what we call absorption. We're going to be focusing mainly at the molecular structural makeup. We shall go ahead and see what are those kind of the monomers and what are those kind of the bonds that are going to be linking these kind of the monomers together to form a very long chain which we call the polysaccharides. And then we shall also go ahead and see the type of the enzymes and the specific type of the bonds these enzymes are going to be targeting to break. And of course, we said that with chemical digestion, we are using molecular features which we call the enzymes. Let's go ahead and get started. Today we have come up with this plate which is going to be containing a lot of different food substances which are going to be giving us a lot of what we call the carbohydrates. On this plate you're going to be having a lot of what we call the big pieces of what we call the sweet potatoes. We are having what we call pieces of what we call the cassava. We are having a big piece of what we call the liver. We also have what we call some fruits and even also what we call rice. Okay, those are the big kind of the substances that we have come up with on what we call on our plate. They are so sweet, they are so delicious. We want to see which kind of the carbohydrates are we able to acquire from all these substances on this plate. The following are some of the carbohydrates that you are going to be acquiring from this plate. The first one that is super very important is what we call starch. Okay, is what we call starch. And then the second one is what we call glycogen. And then the third one is what we call the cellulose. And then also we may have some other kind of the carbohydrate is so minor, which we call the chitin. Okay, so now these are the four main kind of the carbohydrates that you are going to be acquiring from these kind of the foods we have come up on this plate. Okay, now starch is going to be one of the most commonest digestible carbohydrates that we are going to be getting from what we call plants. Okay, so now starch we are going to be getting it from what we call the plants. And then glycogen is a storage form of what we call the glucose in what we call in animals. Okay, so now this kind of the carbohydrates you are going to be mainly coming from what we call animals. Okay, so now that is something which is super very important. So now in this video we want to be taking much little of the time to understand the most common digestible dietary carbohydrate, and that one is what we call the starch. So we are going to be looking mainly on what we call the starch. Now, we shall not pay much attention to what we call this carbohydrate we are calling the cellulose, because the human body is not able to digest what we call cellulose because we do not have enzyme called cellulase. And because the humans are lacking that enzyme cellulase, we are unable to digest what we call cellulose, and this cellulose is going to be helping us in the formation of the bulkiness of what we call the stool. Okay, we are able to form the bulkiness of the stool just because of what we call this cellulose kind of what we call the carbohydrates. Okay, we are going to be paying much time on what we call the starch because it's the most commonly digestible kind of the carbohydrates. Now the question is, we are paying much attention to what we call starch. Okay, so now what are the most common important kind of the molecules that make up what we call starch? That is a big question to ask ourselves. The most common kind of the molecules that make up what we call starch are these long chain of the polysaccharide. You know how this long of the chain is called? We have this chain which is not going to be having any kind of the branch, and we also have this kind of the chain which is going to be having a lot of the branches. So now this kind of the long chain of the polysaccharide, which is a part of the starch, this one here is what we call amylose. Okay, this one here is what we call amylose. Okay, and then this kind of the long chain of what we call of the starch or the polysaccharide, which is going to be having this side chain which is so much branched. This kind of the polysaccharide, part of the starch, is what we call amylopectin. Okay, amylopectin. These are the most commonly digestible kind of the starch, which are going to be a component of what we call the carbohydrates. Okay, I won't take much little over the time, and we enter on this kind of the amylose and amylopectin, and we see exactly the type of the bond, and also we shall also go ahead and see which type of the enzyme is going to be breaking this kind of the bonds. Okay. Now, when you see this kind of this going to be long chain of monosaccharide, there are different kind of the monosaccharides, but we are seeing they are going to be connected to one another. Okay, so now when you see the whole of this kind of this big piece of what we call the monosaccharide, now when you see it's going to be having 6 sides. Now we have this kind of this oxygen molecule on this corner. So now when we start counting on what we call the carbon, on every corner is going to be having what we call carbon molecules, okay? With exception on this kind of oxygen molecule. Now, this kind of the corner is going to be having a carbon, and we label that carbon as carbon number 1. So we are going to be starting to number this kind of the carbons from this side clockwise direction. So now this is going to be carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5, and then we shall have carbon number 6 somewhere there. Okay? Now, when you see this kind of the monosaccharide is going to be linked to this kind of another kind of the monosaccharide, it is going to be having the same kind of the arrangement. Carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5, and then also carbon number 6. Now, what is unique is that we have a connection between these kind of the monosaccharides on carbon 1 and then also what we call the carbon 4. Now, this kind of the bond, which is connecting this kind of the monosaccharide, you know how this bond is called? This bond here, which has been created, is what we call alpha 1 to 4 glyco, alpha 1 to 4 glyco, alpha 1 to 4 glycosidic bond. Okay, that is the name of that bond. This bond is super very important. It is one of the main target of one of the specific enzyme which I will talk about. Okay. So now this chain which we are calling the amylose is going only to be containing only and only alpha 1 to 4 glycosidic bond. There is not any other kind of the bond which is in this kind of long chain of what we call the amylose. Okay. Now when we come to this kind of the polysaccharide part of the starch which we are calling the amylopectin, it is a bit somehow a bit different from what we call the amylose. The first difference is that amylopectin is branched and it is easily digestible than what we call the amylose. Okay. The second kind of the difference is that this kind of the amylopectin is going to be containing 2 types of the bonds. Let's go ahead and see where are these type of the bonds, where are they going to be located. Okay. Now, this one also is going to be now a monosaccharide, and of course, you're going to be having a molecule with the carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5. There is carbon number 6 here. Okay. So now, again, we are now linked to another kind of the monosaccharide. Also going to be having carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5. We shall also have what we call carbon number 6 somewhere here. Now, what is unique is that this kind of the monosaccharide is linked to this kind of the monosaccharide by this type of this bond here. You know how this bond is called? This type of this bond here is what we call alpha 1 to 4 glycosidic, alpha 1 to 4 glycosidic bond. Okay, that is the first bond which is going to be in what we call amylopectin. But when you see this kind of the amylopectin, we are going to be seeing that it's going to be having more other kind of the monosaccharides that are going to be linked to it. Okay. When you come to this kind of these branches, these are also going to be different kinds of what we call the monosaccharides. Now, on carbon number 6, there is this other kind of the monosaccharide which is going to be communicating with what we call carbon number 6. Now, when you see this monosaccharide, this is going to be our oxygen molecule. Now, when we count the carbon numbers in a clockwise direction, this is going to be carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5, and then we shall have carbon number 6. Now, you are seeing that this kind of the monosaccharide is connected to this type of the monosaccharide on carbon number 6 and carbon number 1. So now this kind of the bond which is created between carbon number 1 and carbon number 6, you know how this bond is called? This bond here which has been created, this one here is what we call alpha 1 to 6 glycosidic bond. Okay, that is the name of this bond. It means that this kind of the amylopectin is going to be having two types of what we call the bonds. The first one is going to be what we call alpha 1 to 4 glycosidic bond, and then another one is going to be alpha 1 to 6 glycosidic bond. Okay, so now this also creates a difference between amylopectin and also what we call the amylose. Okay, now having seen now the basics on what we call the bonds which are going to be linking these kind of the monosaccharides together, let's go ahead and see when we put this kind of the substance in the oral cavity, where we are going to be having the first chemical digestion of the carbohydrates going to be taking place. What are those kind of the enzymes that are going to be behind the first chemical digestion of the carbohydrates in the oral cavity? Now, when you come over here, this structure here is going to be one of what we call the salivary gland. Okay, this structure here is going to be what we call the salivary gland. Okay, and particularly we are going to be focusing on what we call the extrinsic salivary glands. This could be the parotid, the sublingual, and then even the submandibular salivary glands. They are going to be producing a lot of this saliva. This saliva is going to be containing a lot of the enzyme. You know how this enzyme which is responsible for carbohydrate chemical digestion is called? These kind of salivary glands are going to be producing this enzyme. We said that these enzymes are like molecular scissors. They aim at moving to break different types of the bonds. Okay. Now that we have seen the types of the bonds in this kind of the amylose and amylopectin, we are going to be using this kind of the enzyme to break those kind of the bonds. You know how this enzyme is called? This enzyme here is what we call alpha salivary amylase. Okay, alpha salivary amylase. It also has another kind of a beautiful name which some other person may call the thiolin. Okay, someone also may call it the thiolin, the thiolin. The pronunciation doesn't matter, but so long as you know what you call the spelling. Okay, so now this kind of the enzyme which you call the alpha salivary amylase, it is the one which is a super very important in breakdown of one of the most important kind of the bond in this amylose and then amylopectin. The question is, this alpha salivary amylase, of these two alpha 1 to 4 and alpha 1 to 6 glycosidic bond, which type of the bond is it going to be targeting? Okay. Now, this kind of the enzyme which we are calling the alpha salivary amylase, is only going to be targeting a bond which we are calling alpha 1 to 4 glycosidic bond. Okay, that is it. Okay, it only targets only one bond which we are calling alpha 1 to 4 glycosidic bond. It has no ability to break alpha 1 to 6 glycosidic bond. It is all that simple. Okay, it means that in the chemical digestion of carbohydrates within the oral cavity, we are only going to be breaking only alpha 1 to 4 glycosidic bond. We do not have that kind of the enzyme that is going to be responsible for breakdown of what we call alpha 1 to 6 glycosidic bond. This enzyme which is going to be breaking this bond is going to be part of what we call the brush border enzyme. It means that we shall have this bond broken when this kind of substance reaches what we call the small intestine. Okay, it's all that simple. It means from the oral cavity to the stomach, there's going to be nothing like the breakdown of what we call alpha 1 to 6 glycosidic bond. It is until this kind of the substance reaches what we call the small intestine, where we shall have what we call the alpha dextrinase, which is going to be responsible for what we call the breakdown of alpha 1 to 6 glycosidic bond. Okay? So now, having seen those kind of the basics, the type of the bond you're going to be breaking and the type of the enzyme that's going to be breaking that bond. Now, in the next video, we are going to now to go ahead and see what happens when this kind of the alpha salivary amylase, when it breaks this kind of the alpha 1 4 glycosidic bond in amylose and then amylopectin. What other kind of the substances are we going to be forming? And of course, at the back of your mind, you should know that you're going to be forming a lot of what we call the disaccharides, the likes of what we call the maltose. We shall even form what we call the oligosaccharides, like what we call maltriose. We shall form even what we call the alpha limit dextrins. We shall even form what we call the sucrose, and all those other kind of the substances which we shall be forming by the breakdown of what we call the alpha 1 4 glycosidic bond within the oral cavity. But what you should know is that the effect of alpha salivary amylase, or which we are calling the thialin, is going to be breaking the carbohydrates in just a very small percentage. Approximately around 10% of the chemical digestion is going to be by the influence of alpha salivary amylase. It is not going to be breaking a bigger percentage of what we call the carbohydrates, okay? Just close to around 10 to 15 percentage of the breakdown by what we call the alpha salivary amylase, okay? All that one makes a lot of sense, okay? So now, having seen all that, let's just have a very quick, brief recap of what we have just said. We said that we are going to be having this plate containing a lot of different food substances. We said that the most common type of the carbohydrates you are going to be acquiring from these substances, the potatoes, the iris, the cassava, the piece of liver, the greens, you're going to be getting starch, which is mainly on what we call the plants, glycogen for the case of animals, so also have what we call the cellulose and even what we call the chitin. We said that the most common digestible carbohydrate is what we call the starch. We said that those kind of the molecules that make what we call the starch, we said we have this kind of this long chain of the polysaccharide which we are calling the amylose, and even also this kind of long chain branched kind of the molecules which we call the amylopectin. We said that these kind of the monosaccharides are going to be joined together by specific type of the bonds. We said on what we call amylose, we are only having this kind of the alpha 1 to 4 glycosidic bond. And then we said that in amylopectin, you're going to be having a lot of the branches, and this explains why it is commonly digestible compared to what we call the amylose. It is having alpha 1 to 4 glycosidic bond. Besides that, it's also having what we call alpha 1 to 6 glycosidic bond. It's going to be containing two types of what we call the bonds. And we said that these bonds are going to be the target of what we call enzyme, which is going to be coming from what we call the salivary glands, mainly what we call the parotid, sublingual, and then submandibular. They release an enzyme which we are calling the alpha salivary amylase, which some other people call the thialin. We said that this enzyme is like a molecular thing that is going to be targeting a specific type of the bond. And we said that this bond is going to be what we call the alpha 1 4 glycosidic bond. It does not have any kind of the effect on cutting the alpha 1 6 glycosidic bond. We said that when all that one happens, you're going to be seeing that alpha salivary amylase has a chemical digestion effect of approximately around 10% of chemical digestion within the oral cavity. Okay, and then besides that, we said that this kind of the alpha 1 6 glycosidic bond, it has its specific type of the enzyme, which is part of the brush border enzymes, which is going to be dealing with it when this kind of the substance reaches what we call the small intestine. Okay, it's all that simple. So now that briefly covers the introductory part on chemical digestion and absorption of what we call the carbohydrates. So TUMNAD, if you think whatever I've talked is making sense, is adding to you, are benefiting, please, you can support him, and that one can be done by a couple of means. You hit the like button, comment down in the comment section. You can share this video to someone else whom you think that this video makes a lot of sense, and please make sure you subscribe. Thank you very much for watching TUMNAD as always. Until we meet next time.